Okay.
Okay, I think I'm going to get started now. Um, Hello, everyone. My name is Josh. I'm a master's student in bioinformatics at UBC. I study under Amrit Singh and Dr. Brad Kwan at HLI at St. Paul's. And today I'll be delivering the UBC Bioinformatics and Statistics Workshop Session 12, which is going to talk about single-cell RNA-seq, cell labeling, and analysis in Python. So I already mentioned this a few minutes ago, but oh, yeah, today for the hands-on example, we're going to use Google Colab. I know in the invitation, it suggested you install Python on your own, but I don't think we're going to have enough time today to troubleshoot Python installations and also Jupyter Notebook installations. So Google Colab is a cloud-based platform that will let us run Python code right on our browser. And so what you can do kind of while I'm talking in this first few minutes is if you could just look up Google Colab, log in with your Gmail account, and ideally you're looking to get to a page like this that will let you kind of upload a notebook. That would be great, and that will allow you to follow along during our hands-on example. Okay, so the learning objectives today, number one is to get a little more comfortable with Python. I would survey the crowd, but I think most people are online right now. But I would assume most people are more familiar with R than they are with Python. So I'm just going to talk a little bit about the differences, and hopefully you can get a little more used to Python. We're going to learn how to use the ScanPy and data libraries, which are commonly used in single-cell analysis in Python. And then we'll also just get more familiar with the single-cell RNA-seq data processing. Oh, I should have done a check. Can I get one more thumbs up that everyone can hear me? Okay, great. Thank you. Okay, so today's format, I'm going to start with a really quick lecture. It'll just be 5 minutes. I'll cover like quickly on the differences between R and Python. I'll talk about a little bit about the AND data library, the ScanPy library, and then a little bit about the 3K PBMC data set that we're going to use in our example today. And then we'll move on to the hands-on example in Google Colab doing the single-cell RNA-seq analysis with the 3K PBMC dataset. So to start, I want to talk about how are R and Python different. Overall, these are pretty similar languages. I think some people find the idea of learning a different language than R a little intimidating, but you'll find as you use Python more, it works in a really similar way. The syntax and style of the code might vary slightly, but all the core ideas about kind of having objects and staying organized are still the same. The biggest difference between the two is honestly the packages that are available. So R tends to be more statistics and biology heavy. So lots of people have written packages that can be applied and do statistical tests or a bunch of whatever you need to do with biological stuff. While Python tends to be a little bit more just generally ubiquitous and machine learning heavy. You'll also find that in terms of the people who actually use the languages, R is kind of niche inside academia and life sciences, but Python is used pretty much everywhere. You can use it in data analysis, and it's also used by like all the big tech companies. I'm sure they use Python, at least at some parts. But overall, knowing both is good. I do a lot of data analysis, and I find I tend to do a mix of R and Python just depending on what packages work the best for me. So hopefully you can pick up a little bit of Python today. And the best way to learn it is definitely just to try to use it is what I would suggest. So if you have an R pipeline, try to transform it to Python. That would be a great first step. Okay. So now I'm going to use two main packages today. And so the first one is ANData. And so ANData is a package that's designed to store annotated data in a nice organized way. It was originally built for use with ScanPy, which is the next package I'm going to talk about, and it's really, really helpful for staying organized. We're going to explore it a lot during the hands-on session. But if you look at this figure on the right here, this is kind of what the structure of an AND data object looks like. So in the middle, this X here, that's like our main data. And then all these obs are annotations about our samples. So if this X was, let's say we had cells as rows and genes as columns, which we will, all the obs are going to be like annotations relating to the cells, and all the var are annotations relating to the genes. And yeah, we'll explore this a lot more. So ScanPy is the other package we're going to use. I think SC stands for single cell, AN is for analysis, and Pi is for Python. So it's built for single cell analysis in Python. It's a Python analog to surat from R. It includes pre-processing, visualization, clustering, differential expression, and more. And it also works exclusively with AND data. I made this little figure here. ScanPy and AND data are like that. They have to work together. And we'll also explore this a lot in the hands-on section. So overall, AND data is for storing neat data storage. ScanPy is for doing your single cell analysis. And finally, I want to talk about the 3K PBMC dataset that we're going to use today. It's peripheral blood mononuclear cells from a healthy donor. It's just 2,700 cells sequenced on Illumina NextSeq, and it's generated with 10x genomics chromium, which is droplet-based single cell. It's a really small data set, which is nice for use in tutorials because it's quick to download, it's quick to run. If you're working with the real world data sets, you probably have more than 2,700 cells. And so when we download the data set, there's already quite a bit of analysis that's already been applied to it. So raw reads have already been processed into a raw cell by gene count matrix, which means alignment, barcode detection, and UMI counting have already been performed, and then empty droplets have been removed. So that was pretty much it for my lecture. We're going to move on to the hands-on example now, and I just want to acknowledge this is heavily inspired by some YouTube and existing tutorials. So I need to move tabs over. First, I'm just going to point out that the dataset we're working with is hosted on the 10x Genomics site. So you can go explore it there if you'd like. Sorry, I need to switch tabs here. And my Zoom bar is getting in the way. Let's drag this down. Okay, so at this point, everyone should ideally be logged into Collab. Has anyone had any issues doing that? Now would be a great time to speak up before I move on. Is there anyone in chat? No? Okay, great. So I'm going to assume everyone has successfully gotten to kind of this page in Collab. Sorry, this is small. I'll make it bigger. It's just the Zoom thing is getting in my way. I need everyone to go to this GitHub page to download a file. So if you just look up GitHub in Google, and this is going to redirect to my page, but if you then go up to your navigation bar and you go github.com/sleung124 and hit enter, we're going to get to this page here. And then we're going to go to repositories. And then there's the UBC Bioinformatics and Statistics resources. Do you want to put the link? Yeah, I will put the link in the, I guess the Zoom chat would work. Oh yeah, that's a lot easier than trying to explain this. Okay, I'll just paste this link in the Zoom chat. Okay. There you go. You should be able to navigate there. And so once we're there, click on Python cell annotation. And there's a file here called single cell RNA python.ipynb. So just go ahead and click that. And download the raw file like this. I'm just going to download it so we can upload it to Google Colab. Give everyone a second to follow along while I switch over to my Google Colab tab. Okay, and then once you're here on Google Colab, you want to go to upload. can browse files. You'll look for your recently downloaded notebook. And we can upload it like this. Okay, and then we should be looking at this. I know that was a lot. Again, is anyone having troubles with this? It's much easier to speak up now than to fall behind and then fall further behind as I move on. Looks like we're good. Is there anything in chat? Nothing. Nothing. Okay, great. So this is Google Colab. So here, this is what the actual file and code that I've written is. And then on the left, you can see there's a bunch of tabs. And I'm going to click this files one because I actually want to see what files I'm working with. You can also see it's allocating runtime. And then eventually that should load and it'll connect to Python 3, which is what we're looking for. And once that's done, we should be able to press play here, and it's going to try to run the code in this cell. And what this is a bunch of, I'm importing a bunch of libraries that I want to use. And this is also something we do in R. Normally in R, you'll do something like library scRNA or something like that. But this is the Python analog, but it's the same idea. This is a bunch of packages that other people have written, and I want to use them. So when I run this, yeah, it's going to say it actually can't find some of these packages. And the reason is because they haven't been installed yet. So if we copy paste this command here, pip install. So pip it's like a repository for Python packages. In R, normally it'll be CRAN or bioconductor. So, and then I'm in the bottom left here, I click on terminal to open up terminal. And then I can go ahead and paste this command in. And you'll see that it's going to reach out to this pip server, and it's going to download all the packages that I just asked it to download. It takes maybe 10 seconds to run.
Do we have it to do it in terminal, or we can do it in the code itself as well? Doesn't.
You can't run it in the notebook. You could put like a %, but I think it's easier to just go from terminal. Okay, so now I can see that that's run, and I've installed these packages that I need. So now if I try to run this import cell again, I'm expecting this to just run properly. There we go. So you can see it gets a little green check mark here, which means the cell is completed. And overall, I think we've talked about R RMD files, or where you can mix Markdown and R. So this IPYNB file it's a notebook file. It's a very similar thing for Python and other languages where I can mix some text and code chunks. I'm going to stop checking in with people to make sure people are following along, but just please speak up if it's going too fast. So in this next cell here, I've defined a function called download and extract data. And so there's this data URL here, which actually matches with this 10x genomics site here. So I'm pointing this data URL right to this data site here, the filtered gene cell matrix. I have to make this small again to move back over. So what this function is going to do is it's going to send a request to the 10x servers being like, can I download this data? And then it's going to, I'm going to extract, it's going to go from tar.gz, which is like a compressed version. But basically when I run it, I'll give it a moment to load here, it should download our data set. And there I can see that I've got filtered gene BC matrices, and then it's got barcodes.tsv, genes.tsv, and matrix.mtx, which is the, I guess, standard output for 10x genomics. So hopefully you have all been able to run this function as well. So now that we have our data, we need to load the data into an ANData object, because if you remember, the ANData package is used for storing and organizing our data. So here, I've written this function, and hopefully you're able to follow along with the code, even if you don't understand what everything means, because I know with Python, it's a little different than R. Like I'm reusing def here to define a function. And that's similar to in R, you might say it would be like load a data function dot dot dot, and then you write the body. It's the same idea here. It's a little bit different syntax, but like idea of functions is still the same. So what happens in this function is it goes and it reads our three files, our matrix file, our barcodes, which are like the labels for the cells, and our genes, and then it loads these into an and data object. And just in general, these, you see, this is like pd.readcsv ad.andData. This is back from up here in our import statements where we're importing the libraries and giving them names. So I'm importing the pandas library as pd and data as ad. But anyways, so then if I set our data path to filtered gene DC matrices HG19, because that's where all of our actual data sets live, I can manually load the A data by calling this function I defined up here. And then I'm also going to use a scanpy built-in function called read 10x matrix, which is going to read the data from the same data path. And so if I run this, oh, I actually have to load this function. There we go. Now, if I run this, we can see that these are two pretty similar and data objects with 2,700 obs, which corresponds to our cells, and 32,738 vars, which corresponds to our different genes. I will note that the scanpy read 10x matrix does a little bit better job than my quick manual one here, so I'm going to just be using this one from now on. And let's see if this works in Colab. So in some things, you could click on that function and actually kind of read what it does, but we can't in Colab, but that's okay. So now we're going to explore kind of how do these and data objects work and how do we work with them. So if I print and data, it's just going to tell me how many observations and vars it has, and then kind of what the vars are, gene IDs. If I print the shape, it'll give it to me in just a slightly different format. If I want to print what the observations are, I can do print adata.obs, and we can see that this is a whole bunch of cell labels. We don't actually have to worry too much about just the random string of letters, but we can just see that there's 2,700 of them, which we expect. If I print data.var, we can see that this is all the genes we're working with. There's 32 something thousand, and all of them have a nice gene name associated with them. And then if I want to access the actual cell by gene matrix, I could do adata.x. But you'll notice it's not printing out like a whole big matrix. And that's because ANDATA stores the gene count matrix as a sparse matrix, which is just more memory efficient than a large dense matrix. But if I do ADATA to data frame, then we can see what our whole matrix actually looks like. And so we have, these are all individual cells, and there's a ton of gene columns, which all have count data. And you'll notice a ton of the counts are also zero, like everything we can see here, which is expected with single cell analysis. Okay, so now I'm going to look at some scanpy functions for kind of how we can do some quality control on this data set. So scanpy has a bunch of built-in functions, and they actually have a doc. So if you just look up scanpy, it'll have a read the docs, and this is where it actually explains what different functions it has and how to use them. Generally, their functions fall into three categories. PP is for pre-processing, tools is TL, and then plotting is PL. We aren't going to use tools very much. Oh, I guess we use tools a little bit today. And we're going to use, I guess, all of these. But you can see in this documentation, it shows kind of all the functions that are available. So it's always good to read that. If you have something you would like to do, scanpy probably can help you with it. So we're going to use this plotting function, so scanpy.pl, and then we're going to plot the 20 most highly expressed genes, kind of just to look at. So I run that. This will take a few seconds to load. And then we can see a plot with the percent of total counts in our 20 most highly expressed genes. So if you have some genes in a particular sample that you're really expecting to be the most highly expressed, this is a quick way to just look at them. And so now I'm just gonna make a copy of our A data object because I'm gonna do a bunch of stuff to this quality control A data, and I always want to be able to refer back to just our base A data, just for this tutorial. So here I'm saying QC A data is a copy of our A data, and then I'm running this pre-processing function called calculate QC metrics on the QC A data. So I run.
What does it do actually? I mean, what do you mean by quality control in a very simple language?
Pardon me?
What do you exactly mean by quality control in a very simple language? Why we are doing this?
Oh, why are we doing quality control? Oh, I will get into this more. Don't worry. But basically, we can see when we run this calculate QC metrics function, it's going to add a bunch of stuff. So this is what we started with. We just had our var as gene IDs, but then when we run this scanpy function, it's added a bunch of obs like n genes by counts and total counts, and a bunch of var. So if we look at these obs, we can now print it out like this. So n genes by counts is basically the total number of genes that have at least one read count in each cell. So for this cell, there's 781 unique genes that have a read count greater than 0. And then total counts is the total number of reads for each cell. And you can see that this varies. Some of them are like 981, some of them go up to 5,000. So later on, you'll see we need to normalize this so all the total counts are the same. And then we can also look at what that scanpy function added to the var. It had added a whole bunch of columns. You can always refer back to the scanpy documentation if you're not totally sure what these columns represent. So now I'm just going to use a scanpy plotting function to visualize some of these. So this is going to make a violin plot of our n genes by count and total counts. So there you go, pretty cool. You could change this however you want to look at different sorts of plots. But what we're actually going to do in this next step is we're going to do some real quality control where we're going to filter out some doublets and dying cells from our cell by gene matrix. So the first thing I'm going to do is I want to add a new column to the var. And if you remember, the var is like the gene names. And I'm going to add a label, which is going to be true or false, whether a gene is a mitochondrial gene. And so mitochondrial gene names start with MT dash. So what this function does is it's going to go through all of these gene names here, and it's going to tell me, does it start with MT dash? And so all the ones we can see here are false. And it's going to add this MT gene column to our var. But then if I filter it for true, we can see that there are, I don't know, 15 maybe mitochondrial genes in our data set. And so hopefully all of you are familiar. If you're not, cells that are dying tend to have a higher proportion of mitochondrial genes. So that's a nice way that we can filter out these poor quality cells. So then I can use this calculate QC metric again, but pass in these QC vars. So this is the scanpy calculate QC metrics function. And then so if I pass in QC vars and I run it, I can see that it actually will add MT genes. So I can get total counts of MT genes and then the percent counts of mitochondrial genes, which is very helpful to work with. And then I'm going to do some scatter plots so we can just look at our data. So here I'm going to plot total counts against the percent counts that are mitochondrial genes. And then I'm going to plot total counts against the number of genes by counts. And in the first one, I'm going to color by the number of genes of counts. And in the second one, I'm going to color by the percent counts of mitochondrial genes. So basically, what I'm looking at here in this first graph, or we can see that some cells have a really high proportion of mitochondrial genes. So here, the percent counts of mitochondrial genes. Anything above kind of 5, which is a bit of an arbitrary threshold, we might not want to use. These cells aren't looking too good, so we might want to get rid of those. And we can also see that the N genes by counts, as a reminder, is the total number of unique genes that have a read in each cell. And so most of these cells are hovering, I don't know, around 1,000, 1,500, but some of them have really high counts of genes, like 3,000 unique genes. So I'm going to assume that those are doublets where they have captured 2 cells in the little droplets and split them both open. And so I want to get rid of those doublets as well. So I'm going to apply some filters where I'm going to filter out cells that have greater than 5% mitochondrial gene counts. And I'm also going to filter out cells that have more than 2,500 unique genes by counts. I should mention there are some more advanced ways to try to filter out doublets or mitochondrial methods, and scRNA-seq actually supports them, but I'm just keeping it simple today. So again, I'm going to make a copy of our A data. And then so this filtered QCA data is I'm basically just going to subset it, and I'm going to look at in the OBS, in the percent count mitochondrial genes, I want to keep everything that's less than 5. So that means anything that's greater than 5, greater than or equal to 5, I'm going to drop. And then similarly here, this is our doublet filter. So I'm going to look in the OBS and I'm going to look at N genes by count. And I only want to keep those that have less than 2,500 unique genes per cell. So when I run this and then recreate our graphs, we can see that we've gotten rid of a bunch of these kind of outliers here. And it's looking a lot more clean and better. And then also, if I print this, I can see that we started with 2,700 cells and we've dropped 62 cells. One of the reasons that this number is so small is we're working with a pretty clean and curated data set. If you were working with your own raw data, you would likely be dropping more cells than this. Okay, does anyone have any questions so far?
Just a quick question about calling doublets. Is that purely based on kind of the average number of genes per counts? Because I guess if you have like a higher average, you would look for things way past that.
Yeah, for the most part here, this 2,500 total count genes per counts is kind of arbitrary. I'm just eyeing up these and saying they're doublets. In practice, I probably wouldn't want to just choose an arbitrary number. There are some methods out there that are a little more rigorous on how you decide what's a doublet and what isn't. So I'm just doing this as an example.
Thanks.
Okay, so now we've done some quality control by getting rid of our dying cells and hopefully getting rid of most of our doublets. Next, we're going to do some normalization and log transform of our data. And so normalization is done to adjust the sampling effects or technical bias in our data because there are lots of things that can affect how many reads we get per cell, like capture efficiency, sequencing depth, other technical confounders. Because if you remember, I'm going to scroll back up. I had somewhere that, yeah, the total counts can vary a lot cell by cell. And if we were going to do a comparison between these cells, we want to normalize all these total counts. So I'm going to use some scRNA-seq functions. So first, again, I'm making a lot of copies of our A data. You don't necessarily have to do that. So I'm going to run this scRNA-seq pre-processing normalized total function, which is going to normalize the sum of all counts in each cell to 10,000. And then after that, I'm going to take the log of these. So I'm going to log transform the data, and I'm going to do log of x plus 1 because a lot of our reads are 0. And if you take the log of 0, you get undefined. It doesn't behave very well. But if you take the log of 1, it just maps back to 0. Okay. So now we've normalized and log transformed our data. Now we're going to do a bit of filtering for highly variable genes. And so scRNA-seq has a nice built-in function for this called scRNA-seq.preprocessing.highly variable genes. So we're going to pass in our and data object. We're going to say we only want to filter for genes that have a minimum mean expression of 0.0125. This number I just borrowed from the tutorial we were using, but basically we want their mean expression to be more than 0. We want their max mean expression to be 3 because genes that have mean expression higher than that are likely more just constitutive genes. And we also want them to have a minimum dispersion. This is where we actually want them to be variable genes. We don't want them to have no dispersion at all. So I can run this cell. And we can see that this adds this column highly variable, which again is just a true or false column. And this is for all of our 32,000 genes. And then we can actually plot these highly variable genes now that we've added this highly variable column. So this is single-cell.preprocessing highly variable genes. This is or scMPy.plot highly variable genes. And so the way you read these plots is here you have the mean expression of the genes, and then you have the dispersion of the genes here. And so everything that's dark is those that we have selected as highly variable. So now we actually need to filter our AND data to get rid of all those genes that that are not highly variable. But you'll notice that I have this line here where I'm saying that our AND data.raw, I'm just going to make a copy of our current AND data. Because later on, when we're going to do some cell labeling, we need access to kind of all the genes, which I'll explain later, or we'll see later on. But when I filter this here, I'm going to filter out a ton of genes, as you'll see. Okay, so now we can see that we started, this is our starting A data, had 2,700 cells and 32,000 something genes. And then now that we've done our quality control on the cells, we decreased it by 62. And then now that we've filtered for our highly variable genes, there's only 2,013 highly variable genes that we're going to use, which is a good step towards kind of dimensionality reduction. But that's still not quite enough dimensionality reduction for us. So we're going to do PCA, or principal component analysis. And I'm choosing to do PCA for dimension reduction because it's relatively quick and easy, and it's going to run fast in this tutorial. But there are many other ways to do dimensionality reduction. You could do non-negative matrix factorization, or you could even do some like really complicated stuff. Like, I'm not sure if any of you have heard of SCVI, which is single cell variational inference, which uses like a variational autoencoder, which is like deep learning, which means you pass in your genes and it trains this like deep learning model that's going to map your 2013 genes down to just like a latent vector with 10 numbers. But anyways, we're going to do PCA here, which scMPy has a bunch of built-in functions for. So again, I'm making a copy of our A data, and then I'm going to call this scMPy.preprocessing.scale function on our A data, which is going to scale to unit variance, which is a requirement before you do PCA. And then I'm going to run this function here, which is actually going to do the principal component analysis. I'm not going to dive into what PCA is here, but it is good to always kind of understand what you're doing. I'm sure there's some very good YouTube lectures that kind of explain how this is going to work. But you can see that this has, again, made some changes to our PCA A data. It's added in OBSEM our XPCA, and it's added some PCs to our VARMs. So what I'm going to do after doing PCA is I'm going to make, this is like an elbow plot, which is really typical for PCA. And scMPy has a built-in plotting function called PCA variance ratios. And I'm going to plot the first 50 PCs. PC stands for principal component. And basically what you're looking at in this plot is on the y-axis, you can roughly interpret as like how much variance is explained by each principal component. And it's a strictly decreasing. So as your principal component number increases, it explains less and less variance about your data. And you want to look for kind of like the elbow in this plot where it's decreasing and then it really starts to kind of level out. And so each of these PCs, if I include them, are mostly just adding noise. So here, I chose principal component 15 right here. That's what I'm going to use. So basically, I'm just going to map our 2,000 something genes down to just 15 principal components, which if I run it, or if I run this cell here, this is what it's going to look like. So for each of our cells, we have a number, which is not very interpretable just reading at it. There are a few ways to interpret PCA, but I'm not going to cover that here. But basically, we've mapped each cell down to a vector or to 15 numbers, which is a lot kind of easier to work with. And so what we're going to do now is we're going to run some algorithms using these 15 principal components. And what we're going to run is we're going to use k nearest neighbors using the first 15 principal components. And basically what this algorithm is going to do is it's going to go through each cell and it's going to find 15 other cells that are most similar to that cell based on their principal components. And after that KNN, I'm going to run UMAP, which is another dimensionality reduction commonly used to plot things. So if I run this might take a little bit to run. I'm also really glossing over what K nearest neighbors and UMAP are going to do because those are quite technical and we don't really have enough time to talk about those, but it's always good to understand.
Can I ask a question?
Yes, go ahead.
So when you were explaining the rationale for picking up only the 15 first, could you please go over that again on why you didn't, let's say, go with only the first 10 PCA? So what was the rationale behind it?
Well, the rationale when you're picking principal components is these are kind of like we have 2013 genes that are describing our cells, but that's basically like a vector or can be represented as a vector of these 2013 genes with a bunch of numbers. But we want to group these cells together and run these k nearest neighbors. But k nearest neighbors will be very noisy and take forever to run if we pass in all 2013 genes. So we want to get latent representations or smaller dimension representations of our cells. So I'm doing PCA here. And so with PCA, it like factorizes your matrix into a bunch of principal components. And each principal component, they're linearly independent and they try to explain kind of variance in your data. And so when we look at this graph here, what we want to do is choose the minimum number of principal components that can explain the most about our data. And so roughly you interpret higher on the y-axis here as it's explaining more about your data, and you can see it has a clear elbow. So with the elbow, we want to grab everything that explains the most about our data and ignore all of these ones that aren't really explaining much about our data. So you could totally choose PC10 here. I chose PC15 just to be a little bit safer, but it's kind of arbitrary what PC you choose here. If the further to the right you go, the more noise you're possibly introducing to your data, but you're also capturing a little bit more signal. Hopefully that makes sense. If not, there are, I'm sure, YouTube videos that could probably explain that a little more rigorously than I just did. You're welcome. So now we can see that after I've run the nearest neighbors and UMAP, it's added even more features to our AND data object. And in particular, in OBSM, it's added this XUMAP. And if I print that, it's a bunch, it's just two numbers that are representing our cells. So we've gone from 32,000 genes all the way down to just kind of two numbers that we can roughly kind of just represent what our cells are. And that's really helpful because we like to make graphs in 2 dimensions. So this could be the X coordinate and this could be the Y coordinate for each of our cells. And then I can plot them using this single cell dot plot dot UMAP. And I can also, that will, if I get rid of this color here, let's see. You guys don't have to do this. I'm just going to try to demonstrate what is happening here. So if I run just this, it's just going to plot our cells on those 2 dimensions. And then you can also add colors to the UMAP, which is really helpful. So if I uncomment that and run this, it's the exact same graph, and here I've colored it by the expression level of this CST3 gene, which I just chose arbitrarily. But what we can now do is we can just do some clustering, which is unsupervised machine learning, where it's going to try to group the cells together. And there's lots of different clustering algorithms you could use. In this particular example, I'm going to do something called Leiden clustering. So it's single cell.tools.Leiden. And so it's going to take in our PCA-A data. And the way Leiden clustering works is it clusters based on the nearest neighbors, which I've already run and is stored in our A data, which is the distances and connectivities. I'm not going to dive into what Leiden clustering does. You can fiddle with this resolution. You can do other types of clustering like k-means. There's lots of types of clustering you can do. It's worth kind of reading about what might be best for your given scenario. But what I'm doing here is this function is going to add Leiden clusters as an aspect of our A data object, and then I'm going to plot the UMAP and color by the Leiden clusters. So you can see that Leiden found 7 clusters. And when you look at the UMAP, it's kind of a good sign that the clusters segregate pretty nicely. There's somewhat clear boundaries. So finally, we've reached the point at the lecture where we can do some cell labeling. And so there's kind of 2 options to look at this. The first one is based on whatever clusters you've found, you can look at some key marker genes and make a dot plot and then try to manually label your clusters. And this is why it was important earlier when we were filtering for the highly variable genes that we also save a raw copy, because a good chunk of these genes actually end up getting filtered out because they aren't highly variable enough, because they aren't expressed in a big chunk of the cells. But we do need them to kind of make these dot plots. So if I run this code, then you can see a dot plot, and I've assigned labels for certain genes are associated with different cell types. And what you could go through here is you could manually annotate each cluster. Like this one looks like it's probably a CD4 cluster because this is the darkest. This one looks like it's probably a CD14 mono. It also has some dendritic cell markers. This is where you want to play around with your clustering algorithm to try to get some nice, neatly defined clusters. But this is the general idea. You can do a dot plot, look at this, and then you can come up with your own label map. Don't trust these labels. I've only done this for demonstration purposes. I'm also just going to copy in 7 because there are 7 clusters. And then I can add this label map as an aspect to our and data object, and then I can plot the UMAP and color by our labels. So that's the normal way you would go about kind of labeling your clusters. But there are some kind of, I thought I would show some other fun ways you can try to label your cells. And so here I'm going to use a library called Cell Typist, and you can look up what Cell Typist is. Basically, they trained these models on a bunch of data sets. Resources, models, on a bunch of data sets, and it's going to look at some key genes and try to make a guess at, it'll go cell by cell and try to guess what particular cell type we're working with. So here, I'm calling celltypist.models.download models, and I'm going to download 2 models that Cell Typist offers. This one is trained on adult COVID-19 PBMC cells. And this one is trained on a bunch of just immune cells. And that website has a lot more detail on what data sets these were trained on, how do the models work. But basically, when I run this, it's going to take a second, but this will have queried the cell typist servers and it's downloaded these two models. So here, I'm going to do cell typist predictions is cell typist.annotate. I'm going to pass in our PCA data. I'm going to tell it what model to use, and I'm going to tell it to do majority voting. So I can run this. And for this last little bit of the lecture, I'm just going to show you a little bit more about how to work with cell typist, just because it's kind of fun. But this will be the conclusion. So here, I have cell typist predictions. Sorry, I'm just reading my code here. Oh, I see. Yes. So when I use majority voting, it's assigning two labels. So one is just the basic predicted labels. It's going through every single individual cell and it's guessing what type of cell is this, and it's giving each cell a label. And so that's what the cell typist cell labels is. And then there's another aspect of these cell typist predictions called the majority voting predictions, where it's also going to look at your neighboring cells and try to kind of aggregate them together. And so if I plot them side by side, you can see that the cell typist individual cell labels, you get a lot of different cell types. But if you do the clusters, you get a fewer cell types. Again, I don't think these labels are particularly good. But this is where you would just experiment with cell typist. And here, I made these predictions using the immune all high model. But if I want to swap this out and use the model that was trained on the adult COVID-19 PBMCs, I could just run this cell, rerun it. It's overwriting these cell typist predictions. And then I can run this again, and you'll see that the cell predictions it comes up with are quite different, just based on the different models you choose. Okay, that brings me to the end of my talk. Does anyone have any questions?
So which approach you would suggest, going with cell typist or going with our own developed coding?
I would suggest maybe you could start with cell typist, but I would always do this manual one on your own. As you can see, I don't actually trust cell typist very much, but it can be a good starting point. And it's also just, I wanted to highlight that there are other methods you can use besides manually labeling things. There are even other libraries besides cell typist out there. You just have to do a bit of a search for them.
Is there an empirical way to define like the optimal number of clusters? Like when you're doing live and clustering?
That's a good question. Empirical, I'm not sure. There are some metrics like adjusted RAND index. I'm going to have trouble naming them off the top of my head. I would have to read into them, but there are some metrics that you can do to kind of estimate how high quality your clusters are. You can also, just eyeballing it is a quick and easy way. Like these clusters, maybe these shouldn't be separate clusters. Maybe they could all be together. But really what I would do is I would run a bunch of different clustering algorithms, and then you can kind of try to come to a consensus. Do they tend to find similar things?
Thank you.
Okay, are there any other questions? I'm actually going to stop the recording.